Hi guys, today we are going to discuss about Teratoma testis, which is the last topic of the series of testicular tumors. And Teratoma testis is basically a germ cell tumor. And let's see the brief explanation without any delay. So we are starting with Teratoma testis. Teratoma testis is basically a germ cell tumor. Why it's called as a germ cell tumor? Because it derived from any three germ cell like ecto, endo, meso. So any three of the germ cell say it is derived, and it is classified into pure germ cell tumor or mixed germ cell tumor. Pure occurs in 2 to 3%. Mixed occur in almost 45%. Mixed with choriocarcinoma or with embryonal carcinoma. It is mixed, and pure germ cell tumor is mainly seen in infancy and in early childhood, and mixed is basically seen in teratoma with malignant transformation, which we'll be seeing later, and also in adults it is observed. So basically it's a heterogeneous tumor which consists of tumor cells which are organoid structures, or those are differentiated cells which are derived from any three of the germ cell layers. Now let's jump to the morphology. In morphology, it is a heterogeneous tumor which is derived from any three of the germ cell layers, and the mixed type of tumor were of as a mixed means other tumor which are infiltrated with teratoma, choriocarcinoma, and also the embryonal carcinoma. And it consists of mature elements, immature elements, and teratoma with malignant transformation. Now let's see first the mature elements ki microscopy. Mature elements means it contains the helter and skelter of differentiated cells and organoid structures, and heterogeneous in nature. So in ectoderm, what structures we see? Ectoderm, first structure we see is the skin. Skin may we see keratinized squamous epithelium we see. Then we see hair. Next what we see is a sweat gland and the sebaceous gland. And also what we see in skin is, along with these elements are the neuroepithelial element, which contains a neural tissue and the glial cells. So basically this is what we see in ectoderm. In endoderm, what we see is the basically bone, islands of cartilage, smooth muscles. Then we see fat, blood, lymphatics. So this is basically observed in the endoderm. What we observe in the mesoderm? In mesoderm we observe gut ka squamous epithelium, intestinal wall. Then what we see is the respiratory tract epithelium like bronchioles vagera, and what we see is the thyroid gland. So this is basically observed. Next what we see is the after completing the mature. Also main important thing is कि mature elements are all absorbed in a fibrous or the myxoid stroma. And also one more thing is all elements resemble the adult tissue. So fibrous and myxoid stroma and resemble the adult tissue. Now we will see it. So in this diagram it can clearly see we have islands of cartilage, smooth muscles which resemble the adult tissue derived from mesoderm. Also the fibrous stroma in which all these have been settled out. Intestinal type of epithelium, which is the component of the mesoderm and endoderm components we can see. Also squamous stratified epithelium, ectoderm component, keratin, ectoderm component. You can see over here nice pictures given. Keratin, stratified squamous epithelium, fibroid or myxoid stroma, islands of cartilage, smooth muscle cells, exact representation of the mature elements. Now next is the immature element. In immature element, a basically definition is it consists of fetal organs or tissues or fetus elements plus mature elements derived from one or two layers. That's called immature element. It will contain neuroepithelium, which is the fetal glial tissue, and also the fetal neural tissue. Plus it will contain mature element like bones, smooth muscles vagera, and yes, immature tissue. Now third one is the teratoma with malignant transformation. This teratoma with malignant transformation is very seen rarely. Basically what is there in this, normal teratoma components are there, like normal teratoma component मध्ये adult tissues are there. Those adult tissue undergo malignant transformation. Undergoes an example, teratoma adult tissue मध्ये bone is there, it will undergo sarcoma. Squamous tissue is there, squamous epithelium which will undergo squamous cell carcinoma, and also teratoma malignant transformation. As we saw, bone, sarcoma, adult tissue into squamous cell carcinoma, and intestine into adenocarcinoma. So this is the basic teratoma testis morphology. Talking about the prognosis of teratoma, it basically all post-pubertal teratomas, both mature and immature, are considered malignant and capable of metastasis, and children mature teratoma are usually benign, and post-pubertal teratoma or teratomas considered malignant and capable of metastasis. Thus, in post-pubertal male, not important to identify immature tissue in a testicular teratoma. For the extragonadal side of teratoma, mediastinum, retroperitoneum, and pineal gland. Yes guys, so this was the basic things about teratoma. We'll continue with now. The video will be continued along with seminoma testis. So yes guys, now we are continuing this video along with a new topic which is called as a seminoma testis, which is the probably last topic of the main plan. So seminoma testis is basically classified into three types: classical or typical seminoma testis. Then next type is the what we call spermatocytic seminoma testis, and we have the aplastic seminoma testis. The three types, we will see all the three types in detail. Seminoma is basically the most common germ cell tumor. That is germ cell tumor, as it occurs about 50% with the preponderance. What is the peak age? Peak age is between 30 to 40 years of age, so that is called as a peak age. Next is the female counterpart of this seminoma testis. Female counterpart is called as a dysgerminoma. It's called as dysgerminoma. And next what we have is the genetic alterations. What are the genetic alterations? This is very important. So genetic alterations, what happens? Two prominent genetic alteration. First is the isochromosome 12p. Isochromosome 12p is there, which leads to activation of OCT3 or 4 and nano transcription factor expression. So OCT3 and 4 and nano. And second is a KIT mutation, and there is a KIT amplification. These are the two important events that takes place. And from where this is about genetic alteration, peak age, common, and from where basically the classical seminoma arise. It arises from the undifferentiated germ cells. Now let's see about the morphology of classical seminoma. Morphology of classical seminoma, first we will discuss about its gross features. So gross features mean what we have is it is understand the mnemonic BRSB, which means bulky, rubbery, solid, and basillated. This is the main: bulky, rubbery, solid, and basillated tumors. How is the size? Size is almost like 2 to 6 centimeter, and tumors are enlarged. So basically, to enlarge 2 to 6 centimeter tumors, which also lead to a enlarged testis. So that's about the size. Now what happens is the cut surface. Cut surface में very important feature that it's a grey white to yellowish tumor which has a lobulated appearance. Also at the same time, it's a sharply demarcated from surrounding normal testicular tumor, and it doesn't show no areas of hemorrhage or necrosis, because areas of hemorrhage and necrosis are common factor which are seen in teratoma testis mixed violet tumor. So there are areas of hemorrhage of or necrosis, sharply demarcated grey white tumor. Let's show diagrammatically. So this is the diagrammatic representation which shows a pale homogeneous tumor grey white on cut section, which is yellowish tan also, and it shows the lobulated appearance. Tumors are 2 to 6 centimeter in size, and they are well demarcated from a normal testicular tissue. Now talking about microscopy, three important facts need to be understood. Those are loosely demarcated capsules. They contain loosely demarcated lobules. Seminomatous cells are arranged in loosely demarcated lobules, which is covered by thin fibrous septa. Alright, thin fibrous septa. Now this thin fibrous septa is basically infiltrated by lymphocytes and plasma cells, or they may be sometimes infiltrated by the giant cells and also granulomas, and the human HCG syncytial trophoblast 15% rare, or also giant cells and granulomas may infiltrated in the fibrous trauma or granulomas. Yes, this is one important thing. Then the talking about a pattern, the tumor cells seminoma are arranged in the form of a nest or sheets or cords, and also they have uniform population of tumor cells. Now classic seminoma tumor is basically resembles spermatogonia. Also single tumor cell resembles a primary spermatogonia. This is a very important, resembles a spermatogonia. Now talking about cell morphology, how with the cell arrangement is done, representation is done. And basically talk about the cell morphology. So cells are round cells or polyhedral cells. Cytoplasm is basically pale or eosinophilic cytoplasm is present. So round and polyhedral cells, pale eosinophilic cytoplasm, and there is basically a distinct cell membrane. This cytoplasm pale eosinophilic, plus it consists watery because it consists of glycogen and lipid. Nucleus is central vesicular with one or two prominent nucleoli. Central vesicular with one or two prominent nucleoli is present. Okay, so basically nucleus, nucleoli, cytoplasm, and what is the basic cell structure. Talking about stroma, stroma of seminal cell, poorly demarcated stroma with thin fibrous septa, and that septa constitute rare feature. And talking about immunohistochemistry, IHC मध्ये VC2, KIT amplification, and OCT3/4 as we discussed earlier, and also there is a isochromosome 12p problem, and we have basically KIT, OCT3/4, and we have one more thing we have is a placental alkaline phosphatase. So this placental ALP, these are present on the plasma membrane, so positive on plasma membrane. What is plasma membrane? How is the plasma membrane? Plasma membrane is the distinct plasma membrane. So we will see now diagrammatically. So as we can see over here, cells are arranged in the form of a uniform seminoma cells, sheets, nest, or cord, which are poorly demarcated, lobulated by a thin fibrous septa. And this thin fibrous septa is infiltrated by plasma cells and lymphocytes. Rarely it is also infiltrated by syncytial trophoblast showing HCG-like action, and infiltrated with giant cells and granulomas. Now talking about the cells, cells are round to polyhedral with pale eosinophilic cytoplasm containing watery texture which contains of glycogen and lipid. Nucleus is central with one or two prominent nucleoli, and also mitosis figures may vary in number. That's about cell, that's about how the lobule is there as discussed over here. Okay, gray white, tumor is homogeneous, gray white, areas of necrosis, thin fibrous septa is present. Next, talking about stroma, the stroma we discussed, thin fibrous septa and infiltration is present. Immunohistochemistry, ALP positive and OCT and alkaline which is alkaline phosphatase, placental alkaline phosphatase is present. So what is the prognosis of seminoma? It's extremely radiosensitive, remain localized to testis for a long time. Metastasis to the lymph node is present, and also we see the hematogenous period occurs late. Now next type of tumor is anaplastic seminoma. So we have anaplastic seminoma. Anaplastic itself says that it is having a wide range of nuclear pleomorphism, cellular pleomorphism. It will be wide. It will have a basic, a very poor prognosis. It will have at the same time you will find a mitotic figure vary in number. There will be a mini mitotic fever, therefore it's called as an anaplastic in nature, and you will find a frequent tumor giant cells. So what are the two characteristics? Many mitotic figures, and we have the frequent tumor giant cell. These are two main characteristics you have to remember in anaplastic type of cells. Now next one is the classical spermatocytic seminoma. Spermatocytic seminoma we'll write. So basically spermatocytic seminoma is both clinically and morphologically a distinctive type of tumors. It is uncommon, occurs in more than 65 years of age, rarely metastasized. So metastasize is rare. It is a slow growing. Okay, so basically it is spermatocytic seminoma originates from the germ cells undergoing spermatogenesis, and also age is above the, so what is the origin? Origin is germ cell undergoing spermatogenesis. Therefore cells resemble secondary spermatocytes. Now we will see the morphology of it. So morphology, it's a basically soft and pale gray tumor, soft plus pale gray tumor it is, and its cut surface shows mucoid cyst. Soft and cut surface shows the presence of mucoid cyst. Mucoid cyst will be seen as the cut surface. Now what are the three major types of cells? Microscopy is dependent upon three main type of cells. So three main type of cells are basically, first is the small lymphocytic cell. Small lymphocytic cells is basically thin rim of eosinophilic cytoplasm. The rim of eosinophilic is a thin rim of basically eosinophilic cytoplasm. Then next what we have is intermediate cells, which will contain a large eosinophilic cytoplasm. That is a very basic difference. And the third one are the large giant cells. Those large giant cells which are scattered in nature, and those may be giants may be a uninucleate or multinucleate giant cells that we don't know. And basically medium sized cells are most common cells seen, which is an intermediate cell, most common cell seen with a round cell, with a round nucleus, cell nucleus. And here it is thin rim of cytoplasm plus secondary spermatocyte resemblance. And also the chromatin of subtumor cell is filamentous in appearance, which is seen in meiotic phase of non-neoplastic spermatocytes, which is known as a spireme chromatin. That is a very important term. What is the prognosis? Prognosis: slow growing tumor, does not produce metastasis, prognosis is excellent. So important is spermatocytic seminoma, prognosis is excellent. Prognosis. Okay. Now yes, so guys, this was an honest effort to explain about seminoma testis and teratoma testis. Previous video before this was about the benign prostatic hyperplasia, which covers actually the all total types of all the male genital system disorders which are including pathology. I hope you like this video and really appreciate the genuine effort and sincere effort. It was very impromptu and undecided to upload this video, but yes, it has been done, and that's all because of your coordination and my constant efforts. Thank you so much. Hope you enjoyed, take the maximum benefit from this video and grab maximum knowledge. Tada, keep tuning.